Paola, for being here, either virtually or in presence. So let's start. So the title of my thesis is Detection of Natural Selection in Recently Admixed Populations. The title is relatively broad by the problem proposed, and that I am trying to solve is now narrower in scope. So let's begin. So natural selection is a process by which organisms adapt to the environment that they reside in. This adaptation um, is viewed in in the phenotypic changes that you can see and are accompanied by changes in their genome. One of these changes could be the changes in allele frequency due to selective pressure on specific alleles. On another note, evolutionary processes take place in populations that are sometimes isolated. And the mixture of two previously isolated populations is called an admixture event. And these events give rise to what is known as an admixed population, consisting of a mixture of the genetic makeup of the parent populations, which with time change independently from the parent populations. In this work, I focused on a special case where populations have evolved in a hierarchical structure and their history can be represented by a phylogenetic tree. Here, population A and B share a common ancestor with X, which in turn shares a common ancestor with C called 0, and where one of the populations suffered a recent admixture event. In addition to that, the type of selection that I studied is directional selection, where the frequency of an allele steadily increases in time until establishment due to homozygotes of the advantageous allele having the highest fitness. The Haberfield statistic will be employed for the detection of selection in this case, while the trimix and other methods will be used for inference of admixture history. So I will begin reviewing some basic population genetic concepts, then review the methods and simulations that I used to generate the results. And finally, I will discuss the conclusions I obtained from my work. So let's dive into population genetics first, with the Hardy-Weinberg principle, which is the most basic principle in population genetics. It states that when a biological population meets certain assumptions, genotype frequencies remain constant through time. Namely, that the population is diploid, that it is hermaphroditic, that it has infinite population size, that random mating is occurring, that there is no immigration, no mutation, no selection, and the generations do not overlap. So when these conditions are met, the allelic frequencies and in turn the genotypic frequencies do not change with the passing of generations. And if we were to understand evolution as changes in allele frequency, there would not be evolution in this system. So the lack of changes in allele frequencies is referred to as the Hardy-Weinberg equilibrium. Now, when working with more than one isolated population, things can get a little bit more complicated. For example, even if these two populations, A and B, are themselves in Hardy-Weinberg equilibrium, the meta population will not be in Hardy-Weinberg equilibrium, where the meta population is the one that contains both populations. So in fact, the genotypic frequencies will not be determined solely by the mean allelic frequency, but also by how different the subpopulations are in terms of the allelic frequency, which is expressed as the variance. We can see that the greater the difference between allelic frequencies, there is an increase of homozygotes at the expense of heterozygotes. And this is commonly known as the Wallen effect. We will call the difference in allele frequencies between populations differentiation. And a useful statistic for measuring differentiation between populations is the FST statistic. The statistics measure how much of the observed variance can be explained by the Hardy-Weinberg expectation of no subdivision. Values close to 0 indicate that there is no differentiation and therefore no population subdivision. So the subdivisions are artificial. And higher values indicate higher differentiation, where the subdivisions have a genetic basis. So we can complicate things a little further. Let's go back to working with a single population, and let's break one of the most important assumptions in the Hardy-Weinberg principle, which let's assume that the population has a finite size n, and the effect of breaking this assumption is known as genetic drift. So here I am representing each individual as a circle with their alleles as ovals and each color representing an allele class. Now the population is not infinite, but has a finite number of individuals n. We will make the simplifying assumption that at the mating stage, all individuals contribute their alleles to a pool of alleles, and the next generation will be composed of a random sample of this pool of alleles with replacement. So if we follow this process multiple times, we will see that eventually one of the allelic classes ends up fixating in the population. The single reason this happens is because the population size is finite, which induces random changes in the allele frequencies and an eventual erasure of genetic variability. So this process is known as genetic drift, and it is a very relevant process in evolution, and one of the main causes for the increase of homozygosity in populations. The mathematical framework behind producing new generations here from random samples of alleles is actually a binomial probability. The probability of obtaining I alleles of type red in a given generation when there are J alleles of type red in the previous generation is given solely by the numbers I and J and by the population size N. So extensive mathematical frameworks on work on drift has been performed before and after the evolutionary synthesis in the 20th century. And this works gave us the mathematical tools to be able to understand how allele frequencies are expected to change through time when subjected to drift. One of the most important frameworks in the diffusion is the diffusion approximation, where when n is sufficiently large, allelic frequencies can be treated as a diffusion process. In this way, the expected value of allele frequency of a given class after t generations will be the initial and arbitrary allelic frequency. And the variance of the allele frequency will increase with the passing of generations, depending on the initial allele frequency and the population size. And with simulations, we can see this in effect, where the mean allele frequency over replicates, shown in red, stays roughly close to the initial allele frequency that is shown in blue. And the variance of the allele frequency increases with time, and it is higher for lower values of population size. This is when dealing with a single population, right? But what happens when we try to deal with hierarchically structured populations, like we see in this tree? What are the expected allelic frequencies for each population, A, B, and C? What are their variances? Do they show a covariant structure due to their shared history? Well, first, we can treat each population separately under the same previous framework. So the expected allelic frequencies will be the initial allelic frequencies. In this case, the allelic frequency of the most recent common ancestor is denoted as P0. So what about variance and covariance? The variance can be treated similarly, but remember that the variance depends on the time that has passed since an initial condition and the population size. So by making a few simplifying assumptions, we can encapsulate both these parameters in a single parameter, which we'll call delta, or a drift coefficient. In the context of hierarchically structured populations, this drift parameter will reflect the amount of drift that the population has been subjected to, as a combination of population size n and the time in generations under drift. These drift parameters will be reflected as branch lengths in this phylogenetic representation of splitting populations. So if we use vector notation for convenience, the vector of expected allelic frequencies of A, B, and C will be P0. And the covariant structure of the allelic frequencies will be given by the drift parameters and the topology of the population phylogeny. The covariance of two populations will be the drift parameter that corresponds to the more recent common ancestor. In this case, for A and B's, the recent common ancestor is X, so the covariance is the drift parameter associated with X. The covariance matrix will be then the matrix containing the information about drift and topology multiplied by the expected variance of initial allele frequencies. This allows us to treat the vector of population allele frequencies as a multivariate normal distribution with mean vector P0 and covariance matrix V. Now, what if there has been an admixture event? How can we extend the previous framework to accommodate for an admixture? This can be done by including an admixture parameter M, which is a ratio of the contribution of allelic frequencies from one population to the other. In practice, this results in a slightly modified covariance matrix, where a new correlation structure is induced because of the admixture event. We can then treat this the same way we did before, where allele frequencies follow a multivariate normal distribution. This treatment of the covariance structure was under admixture event, was developed by Pickrell and Pritchard 2012, which is in turn built upon a model of SNPs under drift by Nicholson 2002 with its roots in Cavalli's Forza 1975. Now, for the sake of context, allow me for a small digression in the topic of genetic linkage. Say we have two loci with two alleles in both. Say we have two loci with two alleles in a single chromosome. So with a recombination probability R between them. In a population, there can be all different four combinations of alleles, which we will call haplotypes. And these haplotypes will have different frequencies that will depend on the allelic frequencies, but will not be quite equal to the product of their frequencies. They will not be independent. This non-independence can be summarized with the statistic D that expresses the departure of the haplotypic frequencies from a state of independence. In a single population, this statistic will decrease exponentially with time depending on the recombination rate, which will itself depend on the distance between loci, among other things. This non-independence of genotypic configurations will cause a correlation structure of alleles across the genome. And in nature, there will be blocks of the genome that will show high allelic frequency correlations. These blocks are known as linkage disequilibrium blocks. Now, so far I have exposed the dynamics of allelic frequencies under different models where no selection is present. In other words, under neutrality. Let me give a brief overview of how selection can affect allelic frequencies. So let's focus on a locus that has two alleles, A1 and A2, and let's suppose that A1 is advantageous. This means that A1 provides individuals with an increased fitness compared to the A2 allele. In this case, the most fit genotype will be the homozygous A1, followed by the heterozygous A1A2, and the homozygous A2. So with this scheme, which is formally called selection with incomplete dominance, we can distinguish two processes that involve selection, but differ in the moment that selection starts to act with respect to the advantageous allele. The first is a hard sweep, where initially there is no advantageous allele, but eventually it is introduced into the gene pool by mutation or some other process. This will cause an immediate rise in allele frequency for A1. Contrast this with the soft sweep, where the A1 allele is not advantageous at first, and it already exists in the population with a given frequency. Once the environment imposes a new selective pressure that makes A1 advantageous, it will begin to rise in frequency through the generations. And these two examples will leave different fingerprints in the genome. In a hard sweep, the advantageous allele occurs in a single haplotype, and due to linkage, nearby alleles will rise in frequency as well. This usually leaves a distinct mark on the genome, like we see in the bottom of the slide. When heterozygosity is measured along the chromosome, a decline is observed when moving towards the locus under selection. In the case of a soft sweep, once A1 enters a selected regime, it is already present in multiple haplotypes. This will cause the haplotypes to increase in frequency, leaving a more subtle mark on the genome when measured with heterozygosity. So the methods used in this work can be roughly separated into two categories: covariance structure estimation and detection of selection. All of these methods will use genotypic data in the form of SNPs. Let's say we have three populations, A, B, and C, that have evolved in a tree-like fashion, and that it has also suffered a recent admixture event shown in red. So the objective is to estimate this unknown demographic history with only genotype data from the populations in the leaves A, B, and C. And knowing this demographic history, we can get an approximation for the covariance structure of the genotypic frequencies. First, if we actually know in detail what is the demographic history, we can express the covariance matrix like we did with the examples before. So using the drift parameters and the admixture coefficient, we can compute a theoretical covariance matrix under the simplified model. And this is, of course, not possible for natural populations, but it is possible for simulations where we specify all of these parameters. This theoretical matrix will be used as a gold standard when comparing different estimation or covariance methods. So another method for computing the covariance will be the kinship method developed by Bonhomme et al. 2010 in their FLK paper. So using genotypic data, Reynolds distances are computed with SNP allele frequencies, and a neighbor joining tree is constructed. Then an F matrix is calculated directly from the branch length of the neighbor joining tree, which reflects the covariance structure of the populations. So another method, which we will call empirical, and here is the same tree, uses the F matrix computed with the kinship method to estimate the ancestral frequency P0, where P0 hat is the best linear unbiased estimator of P0. Then for each locus, a Q vector is computed from the allelic frequencies and the estimated allelic frequencies. And this vector can be interpreted as a vector of allele frequencies normalized by the ancestral allele frequency. So the covariance matrix estimated by the empirical method will be the sum of the order product of the Q vectors over all loci. Well, so far, none of the previously mentioned estimation methods takes admixturing into account in an explicit manner. So the software Trimix does and computes a covariance matrix by explicitly modeling and estimating migration. The goal of the Trimix method is to estimate W, the theoretical covariance matrix that we saw previously. And this is done by treating the admixture tree as a graph G and computing a maximum likelihood estimate for W for a given graph G. So the general idea is to maximize the likelihood of a covariance matrix for a given admixture graph. So because it is computationally infeasible to enumerate all possible admixture graphs, Trimix relies on a heuristic, which consists of, first, computing a maximum likelihood tree from all the allelic frequencies, then inserting a number K of admixture edges with specific criteria, and finally optimize the branch lengths and admixture weights by maximizing the likelihood. And this is done iteratively until convergence to a minimum. So we have these four methods to express or estimate the covariance structure of a number of populations from their allelic frequencies. Now we will review the methods used to detect selection using genotypic data. There are different classes of methods used to detect selection. One of the most popular is to define a statistic and measure it throughout the genome, looking at loci with extreme values of the statistic and then mark them as candidates for selection. And this approach underlies the assumption that every polymorphism is subject to demographic effects, but only a few will also be subject to selection effects. One of the first methods developed with this concept in mind was the LK test. So for n populations, the LK statistic at a locus is the ratio of the locus, FST, and the mean FST multiplied by n minus 1. So under the null distribution of LK, the locus is neutral. And under the alternative distribution, the locus is under selection. This test assumes that allele frequencies are drawn from a normal distribution and that most loci are only subject to demography. Under the null, the LK statistic follows approximately a chi-squared distribution with n minus 1 degrees of freedom, being n the number of populations. But the LK statistic was heavily criticized when it came out. Among other reasons, it could not correctly handle tree-like populations, and it would give inflated values for populations that had been under bottlenecks. So in 2010, Boname et al. Developed the FLK test, which extends the LK test to account for the hierarchical structure of populations and the variability of population size. The FLK statistic takes this form, where the variance is F, which is the matrix estimated in the kinship method, times P0, 1 minus P0, where P0 is the best linear unbiased estimator of the ancestral allele frequency. So first, it is necessary to estimate F and P0, and the underlying assumption is that the allele frequencies follow a normal distribution depending on the estimated ancestral allele frequencies. So the FLK statistic follows under the null a chi-square distribution with n-1 degrees of freedom. Also, the FLK test can handle multiple alleles, but the math here is a bit more complicated, but the concept is the same. When using multiple alleles, the FLK distribution follows a chi-square distribution with n-1 times a-1 degrees of freedom under the null, where A is the number of alleles at the locus, of allele classes at the locus. So now enter the HAP-FLK test. This method was developed in 2013 by Fariello et al., and it extends the FLK test to include haplotypic information to detect selection. Now, for the sake of brevity, the rationale is to cluster haplotypes in K clusters using the fast face algorithm, which paints haplotypes according to the similarity of allele classes along the genome, like this. Then treat this cluster as alleles and compute a modified multi-allelic FLK statistic. So the actual computation of the HAP-FLK statistic has a few more caveats. So if you're interested, I recommend the paper. So if we call Q the K times N vector of allele frequencies, HAP-FLK is computed as follows. And it follows a distribution proportional to a chi-squared with n-1 times k-1 degrees of freedom for K clusters. Then p-values are computed for a chi-square distribution, and after multiple tests and correction, it can predict loci that have been likely under selection. So the main idea of my work is to evaluate the performance of the HAP-FLK statistic when provided with different covariance matrices. Theoretical being the gold standard, kinship being the previously published method, empirical, which is a new method not taking explicitly into account admixture events, and trimix that explicitly models admixture events. First, I studied the empirical behavior of the different estimates of the covariance matrix under the null distribution, and I named this section calibration. Then I evaluated and compared the power to detect selection for the different methods, and I called this section evaluation. I performed these studies using simulated genotypic data, as it allowed me to know the exact demographic history of the populations. I used the forward population genetic similarity SLIM, which allows to simulate complex scenarios with population splits, migration events, and selective regimes. And it can be programmed to output genotypic data in conventional formats such as PCF. So now for the calibration scenario, I performed two simulations with different levels of complexity. The scenario A consists of 4 populations with a tree-like history, and P1 is considered as an outgroup population. An admixture event takes place where population P4 receives an admixture pulse from population P2. And scenario B is slightly more complex, but similar, with 2 additional populations, P5 and P6. Still, P1 is considered as an outgroup population. Now, following the model that I showed previously, the covariance matrix of A can be expressed in terms of the drift parameters and the admixture coefficient. And similarly, the covariance structure of scenario B can be expressed that way. In all cases, when applicable, the selection regime takes place as soon as the admixture pulse takes place. For the evaluation scenario, I simulated the following tree, where an admixture event takes place between P2 and P4 to give rise to a new population, P5, and P1 is an outgroup here. So here is the corresponding covariance matrix with the given drift parameters and admixture coefficient. And the selection coefficient S is either 0 or 0.1. The admixture coefficient is M is either 0, 0.3, or 0.7 for evaluation. For each set of parameters, around 100 replicates were simulated for calibration and 1,000 for evaluation. And in all cases, population size was remained constant at N, at 1,000 individuals. Sorry. Now I will show the main pipeline for computation and handling data. The pipeline was managed with the domain-specific language Nextflow for reproducibility. So the first step was to specify the demographic scenario to be simulated. This includes the tree topology, the drift parameters, and the admixture and selection coefficients. This was done by writing specific SLIM scripts that output genetic data into VCF files, which are then converted to the BED format. And the genotypes are filtered for low minor allele frequency. So the different covariance, matrices are estimated using these different methods that we already mentioned, but for the theoretical, I specified the tree topology, the migration parameters. Then the HAPFLK software computes both the kinship and the empirical matrix. And for Trimix, well, it was used the Trimix software. In this case, the genotypic data is filtered in order to retain loci that are under linkage equilibrium. So the genotypic data from the simulations is provided to HAPFLK together with the corresponding covariance matrices. And this will generate a database that contains the position, the replicate, the covariance method used, and the HAPFLK value for a given locus. So now for the results, as I mentioned earlier, they are separated into calibration and evaluation. In the calibration step, I obtained the empirical distribution of the statistic under the different covariance matrices. I assessed their fit to the theoretical expectation and determined the best way to evaluate statistical significance. So let's begin with the scenario A. Here we see the densities of HAPFLK using the different covariance estimation methods. And we can see that all distributions look similar, both in the presence or absence of admixture. We can note that for the Trimix method, the values are actually scaled to the right and they show a higher variance compared to the other methods. And this is expected. As we discussed earlier, the estimation of W under Trimix includes a nuisance parameter, which is the expected variance of the ancestral allele frequencies. And this is deliberately included in the Trimix model, but is missing in the other estimation methods, which causes a scaling of the computed HAPFLK values. So in the case of the scenario B, we can see the same phenomenon happening, where the Trimix values are scaled to the right and they show a higher variance compared to the other methods. So here we can identify more clearly in the example where M equals to 0.3 and S equals 0.1. We can identify the linear relationship between the values of HAPFLK. In particular, regarding the Trimix method, we can see that it is linear, but the slope is not close to 1. The scale is different. Now, we want to know how well these distributions fit to the expected chi-squared distribution. So I performed a visual assessment using QQ plots for the different scenarios. I compared the quantiles of the HAPFLK distributions with the quantiles of the theoretical chi-squared distribution, and points within the 95% confidence interval band would imply a good fit to a corresponding chi-squared distribution. In this case, in the absence of admixture, we can see that the fit is relatively good, with maybe the theoretical estimation being a little bit off. But if we see with m equals 0.3, I mean, in the presence of a mixture, we can see that the estimation is, sorry, the distribution of HFLK is actually a little more heavy-tailed compared to a theoretical chi-square distribution. So for a scenario B, we can observe a similar trend with a slightly better fit in the presence of admixture, except for the kinship estimation, which we can see that it shows a heavier tail. Now, we know that using quantiles, the fit to a chi-squared is relatively good. So we should be able to compute p-values from this distribution, right? Right? Well, no. Remember that I mentioned that the HFLK statistic is proportional to a chi-square distribution of the given degrees of freedom. So this means that it is a chi-squared in shape, but with a different empirical value for the degrees of freedom. We can see this when we plot all densities in the same plot. Here, empirical, kinship, and theoretical are superimposed, and is that peak to the left, and trimix is scaled to the right. And here we can see that the black curve represents the density for a chi-square distribution with n-1 times k-1 degrees of freedom. So how can we compute p-values in the setting? One way is to perform a quantile regression and rescale the HFLK values that we produced. So this is done by computing the quantiles of a theoretical distribution and the HFLK distribution and performing a robust linear regression of the theoretical quantiles onto the observed quantiles. Then the new rescale values come from multiplying the old values with the regression slope and summing the intercept. So with this rescaled HFLK values, we assume a chi-square distribution with the given degrees of freedom and compute p-values. As we can see, we obtain p-values that are uniformly distributed under the null, which is good. This is what we would expect if the statistic behaves well. So now, in the next section, with a different scenario and more replicates, I evaluated the power to detect selection using the different covariance matrices. Here we can see the topology, and in this case, the admixture coefficient is either 0, 0.3, or 0.7, and the selection coefficient is either 0 or 0.1, where S equals 0 gives us the null distribution of HFLK under the null hypothesis. So how do we compute the power of this statistic? Remember that the null hypothesis is no selection, with the alternative hypothesis being that the locus is being selected. So following this, under selection and neutrality, I computed the genome-wide maximum of the statistic for each replicate, S-max for short, and obtained the empirical null distribution and then empirical distribution under the alternative hypothesis. So with these distributions in mind, let us compute the power of the statistic. We will define P as the proportion of replicates under the alternative distribution where S max is greater than Q alpha. And Q alpha is the 1 minus Q alpha quantile of the S max under the null. Then P is the power of the statistic at the type 1 error alpha. In other words, if we divide the null distribution of X S max at the 1 minus Q alpha quantile, and we take the number of replicates to the right of this value in the alternative distribution and divide it by the total number of replicates, we get the power P for the statistic. So in this way, I computed the power for the different covariance estimations at different values of type 1 error. We can see that in the absence of admixture, all methods perform relatively similar, with the exception of trimix, which is the one that shows the worst performance. At low type 1 errors, we can see that the performance is relatively bad. In the case of an admixture coefficient of 0.3, there is a clear difference in power, with the theoretical matrix being the best performing, followed by the empirical, the trimix, and finally the kinship matrix. Again, the power for low alpha values are very low, except for the theoretical matrix, but this is not obtainable in natural circumstances. At an admixture rate of 0.7, we can see that the power decreases dramatically for all but the theoretical matrix estimation, and this is increased at low type 1 errors. So the the ROC curves that we have seen so far are computed using various replicates that have suffered both hard sweeps and soft sweeps, and things in between, which are determined by the allele frequency of the adaptive mutation at selection onset. So a natural question to ask is, well, how does frequency at selection onset affect detection power? Here, with m equals 0.3 and s equals 0.1, we can see the distribution of frequencies of the advantageous allele in the different populations at the onset of selection for an aggregate of the replicates. So the population that is actually under selection is P5. And what I did to evaluate the effect of frequency on detection power is group replicates by frequency at selection onset, like this, and then compute the power for each group at a type 1 error of 0.1. So now, the results are summarized here for the different values of the admixture coefficient M, and in all cases, we can clearly see that as the frequency at the onset of selection increases, the power decreases dramatically. In the absence of admixture, all covariance estimation methods behave similarly, with trimix being the worst performing method. When m equals 0.3, the theoretical matrix performs the best, followed by the empirical, the trimix, and the kinship matrix. And when m equals 0.7, we can see a drastic decrease in power along all frequencies, with the empirical method performing the best at good working frequencies. This shows that all of these results shows that the empirical method is the best suited for detecting selection and shows the best power, at least in this case. So this decrease in selection in detection power with the increase of frequency has already been showed and described in HAPFLK by Fariello et al. And I reproduced these results and extended them to include other methods of estimation of covariance. So now concluding. In this work, I evaluated new procedures using the HAPFLK method, namely the empirical covariance matrix and the trimix model matrix. I was able to reproduce previous results regarding HAPFLK and the dependence of power on frequency at selection onset. When using the empirical covariance matrix and under favorable conditions, I was able to confirm that reliable detection of selection in recently admixed populations is possible with the HAPFLK method. And one particularly interesting result is that the best performing method, the empirical covariance, is model-free, and it is included in the latest release of HAPFLK. This is particularly convenient as no intermediate steps are required when contrasted with using trimix. So similar conclusions had already been pointed out in other research, especially in Rifollo et al. 2019, where there is a trade-off between interpretability and power, where you can get an interpretation of the demographic history at the expense of power for detecting selection. So now, what could we do from here regarding future work? Well, what I think is that I should benchmark my results or at least the method against other methods to see how well it performs. I should also test it on empirical data and try to reproduce previous conclusions with empirical data, and maybe also include more methods for estimating covariance structure. Recently, Molloy et al. Developed a framework for improving the results of the trimix algorithm by moving the local optimum to a lower optimum. So it would be interesting to implement this algorithm in the same framework as my work to see if trimix improves. And that's it. So any questions? I don't know which microphone we should use.
Yours, which?
Okay.
Sorry.
I'll turn my speaker on too.
You're the president. Yeah. Turn your microwave.
So thanks for the presentation. We're going to ask questions, beginning with the members of the jury, okay, of the panel. So maybe Paola, if you want to, Fernando, if you want to start with questions or comments, take the floor.
Thanks a lot. I think it was a very, very nice presentation, very nice thesis. It's very clearly stated everything, and like it's a very thorough evaluation of all the different assumptions that are going into these methods. I have several questions. I don't know if you guys, do you want me to like go through them all or just start with one and then give the word to somebody else? How do?
No, you can do them all, and then we will pass through Paola and then Enrique.
Okay. I guess the first one is, I mean, I have some suspicions about this, but why do you think the empirical covariance matrix is so much better than the other two matrices? Like what do you think you're capturing there that is not captured in the other approaches?
Yeah, so I guess the reason for that is that when you use the empirical covariance matrix, you're actually like including all possible information. And when you use a method like tree-mix that uses maximum likelihood to estimate a covariance matrix, you're probably leaving some information out. And also tree-mix possibly, I mean, when estimating with tree-mix, you specify one migration edge, and this is added in different places according to the heuristic. It can go to a local minimum, to several minimums of equal likelihood, and also the tree-mix method was performed like blindly. It was not supervised. Like I gave them the data and they produced a matrix and that was it. So maybe with a little more guidance, it could give better results. Yeah.
Okay, another question, and this one I don't have I mean, it's an honest question, but why, so under the condition where you have high migration, like I think it was 70%, you show that the theoretical matrix is still doing quite well, but the empirical one is not. And that was somewhat confusing to me. I would expect why the other two would not do very well, because they're sort of making sort of low admixture, low frequency assumption, like assumptions that admixture is infrequent. But why do you think the empirical one is not doing well under that situation?
Yeah, I had the same doubt. I have a few guesses, but I'm not really sure. The first one is that, first of all, when estimate with all of these estimation methods, basically what you do first is construct a population tree, and then from that compute the covariance structure, whether there is explicitly accounting for admixture or not. So when you construct this tree, the final allele frequencies are very important, because this will give the estimate for a maximum likelihood or neighbor joining tree. So when the admixture ratio is really high, probably the admixed population is located with a common ancestor on P4. Let me show to explain myself a little better. So Yeah, here. So we can see here that you're asking for the case where M here is 0.7. In this case, P5 for when constructing a tree would be put with P4 at first. And it would seem as if the most recent common ancestor would be between P5 and P4 would be a node that is not B. So that could affect the estimation. I think that is one of the main reasons. I was thinking about another one, but I forgot. Maybe I can tell you later.
Cool. Now the question is, so you have as you very well explained, you have a Wright-Fisher model, and then you approximate that using a diffusion approximation, and then you do this multivariate normal approximation to the diffusion by matching the first two moments. This is more of a broad question, but under what conditions do you think this multivariate normal approximation is appropriate to the process that you're trying to model, and under what conditions do you think it might not be appropriate?
It is clearly not appropriate. It is clearly not appropriate for small population sizes because when the population size is large, the process actually works like a random walk, like a Brownian motion. So it can be approximated through time with a multivariate normal distribution. But when n is small, I say, I don't know, below 1,000, but I'm not sure about the cutoff. It would not work really well. It would also not work well where there is a lot of mutation going on, because it would introduce new mutations that well, before that assumption breaking, the next assumption that would break the normal multivariate distribution would be the extreme frequency alleles. If you have extreme frequencies, you don't have a normal. You have like a cut normal distribution. So following that, if you have mutation, it would also break the assumption. I think those are the main ones.
Good. And then can you tell me under what sort of populations we're modeling here? We're modeling them here as either tubes or mixtures of tubes. Under what sort of real life scenarios do you think these assumptions that we're making about the hierarchical structure of populations might not hold up very well? What are sort of demographic conditions where the application of these sort of tree-like structures might not work so good, and why do you think that is?
Possibly, if I understand your question correctly, you're asking in what kind of real life situations this would work or would not work. Is that correct?
Yeah.
Okay. So where there is continuous migration, I think it would break up. Like, even if there is a hierarchical structure, if there is a gene flow between the populations and the leaf nodes, the covariance structure would be much more complex. So you would have to either, I don't know, perform some more sophisticated demographic inference methods or something like that. So I don't know, in human populations in general, there's a lot of admixture. There's not much isolation, so it would not be applicable. In the case of some geographically isolated populations, I guess it could work, like plants or birds in islands, for example, or some other island organism. Maybe birds not, because they show a high gene flow. But for example, in livestock populations, it is very common that we see these hierarchical structures because we select for different traits, and we isolate the populations on purpose to exacerbate some traits. So in general, in livestock or any artificially selected population species, I think this could work. Yeah.
And so in your simulation scenarios, you're testing a scenario of like strong selection. And I guess I have two questions regarding that. Like one, you mentioned at some point in your thesis that like sort of if you're looking at sort of like a small region and you're trying to estimate the covariance matrix, and that region is affected by strong selection, that might bias your inference. What would you do in those scenarios? What would you do to sort of assess how much selection throughout the genome is affecting your estimation of the covariance matrix? And the second one, you also talk about soft sweeps and sort of other modes of adaptation. I guess, have you looked at sort of the power of these tests? Well, I guess you have this plot of the starting allele frequency under different scenarios, right? So would you recommend to only apply these methods when you're looking for strong selection, or do you think they could work for soft sweeps as well?
Well, for the first question, what I would do to better the results when they could be biased by using a short genomic region, let's say, I would try to simulate several like chromosomes, for example. In my work, I simulated a single chromosome. I didn't mention that. I simulated a single chromosome of 10 megabases. So in real life, what you get is several chromosomes, and maybe some of them are under selection or whatever. So that would be a way to get better results regarding that situation. And for the second question, I'm not sure. I guess you could see. I mean, it's not very computationally expensive to run this, so you can run it as a like for preliminary results. For selecting soft sweeps. Soft sweeps are really hard to detect in general. There has been recent work regarding that, but it is still a thing that is not easy, at least with like atemporal, or let's say spatial data. In the case of temporal data, if we have ancient DNA or whatever, we could actually see the rise in allelic frequency and some other haplotypic information. So that could be possible, but not at least in this setting.
I think that's most of the questions that I had, so I'm going to give the word to my companions. But yes, very, very nice work.
Thanks, Fernando.
Paola?
Okay, hello, thank you very much. First of all, I want to thank Gastón for inviting me to his thesis. I really appreciate to read it. I enjoy it. I can see a lot of work in several domains, I say, of your simulations, your effort in programming and in different languages and combining the different worlds. And also in the statistic area, there's a lot of interesting problems, so I really appreciate it. And also the presentations was very, very nice. So I have some questions also. I was a little bit, I don't know how to say, surprising about this scaling difference and the percentile and the quantile regression that you have to do. I don't know if I really understand the original problem. Do you think that the approximation to the chi-square distribution is proportional, or is an approximation? I don't know. I didn't read the original work about the theorem behind this result, but I didn't understand this approximation to this is chi-square, or if it is proportional. And then maybe there's two possible sources of this difference. Maybe one is in one moment you say you choose the B matrix by an identity matrix. So I wonder if this step is maybe a source of difference. And then also behind all of this is the number of clusters that is detected by the Hayden-Markov process. And so maybe if you, I don't know, if maybe you with the quantile detect the number of clusters, then try to, with this number of clusters in the Markov process, maybe they will change the distribution or the fit to the distribution.
Yeah, exactly. Well, let me move to a slide so I can explain my answer better.
Perfect.
So yeah, here is the how we compute the HAB-FLK distribution.
Mm-hmm.
Basically, what I understood from your question is your question is that is it approximate? Is follows? Is it proportional? Well, both. It is both approximate and proportional. First, it is an approximation because here in the multi-allelic FLK test, it follows approximately a chi-square distribution. So if you perform the same statistical, if you perform the FLK test with real multiple alleles, you get an approximately chi-square distribution with n-1 times a-1 degrees of freedom.
These approximations have the origin in the assumption of normal distribution for the flex.
Exactly.
That is okay.
Yeah, that approximation has the origin in the just like
in the normal, yes.
test, for example, it makes the same assumptions that they are drawn from a normal distribution, etc. So it's the
That's
same.
perfect.
So then it is proportional because of what you were saying, the B matrix. If you can here the TFLK is computed using a B0 matrix that includes information from the ancestral allelic frequencies. But here, when you compute the HAB-FLK statistic, you use an identity matrix. In practical terms, this was performed because the B matrix was unstable when using clusters. So you couldn't, was unstable in terms of it wasn't, yeah, you couldn't invert the resulting matrix. So,
okay.
So yeah, that in practical terms. In more of theoretical or speculative perspective, I guess, the B matrix in this case would not be a real matrix because you don't have ancestral clusters, because the clusters are actually just a construction. They are not real. They are not haplotypes. They are not alleles. It's just like a way to capture haplotypic information. So the B0 matrix in this case would not be something that would work conceptually. So I think that is why that is why you can see the instability in those cases. I am not sure how the Hayden-Markov model affects the distribution of the HAB-FLK, how the choice of clusters affects it. But yeah, that's it.
Okay. Okay. Now it's clear. Thank you very much. And then the way to improve these results is with the fitting of the quantiles.
Exactly. You force the empirical distribution, let's say, of the HAB-FLK to have the degrees of freedom, to have the mean and variance of a corresponding chi-square distribution. So you keep the shape, but you do a linear transformation. So I think it's equivalent.
Yes. Maybe with this fit, you can estimate a new number of clusters and see if with this number of clusters, because it's the degree of freedom, you have n-1, so you can get k, and see if with this new number of clusters, the Markov model is performing well of the number of, if it's a reasonable value for the number of multi-allelic or haplotypes or the things. But it's just an idea.
I think that I could look into that. Yes.
Okay, and you try to see. You see that you try to estimate with a normal approximation, and you see that it was not good. But you try it with the scale parameters, a normal distribution with the scale parameters.
No, I did not. That's
Okay.
a good question. I don't think it would change much.
No, but
yeah.
I didn't change. I think it will not change much with respect to the chi-square distribution fit that you did. Okay.
Yeah, maybe just a little note here. The normal distribution could work better if I had simulated more populations as well, because of when the chi-squared, as the chi-squared distribution depends on the number of populations, the degrees of freedom depends on the number of populations. When the number of populations increases, you can do an approximation to a normal distribution. So maybe if working with several more populations, a normal distribution would perform better, but I didn't test that.
Okay, thank you very much. I think I have minor comments about some formulas or sub-index. The vectors were sorted in a way that was a little bit confusing for me. I expected PA, PB, PC, but it was PB, PA, because I think this is the number of the leaves of the tree, you know. And so it was a little bit confusing, but it was my fault. And then about the results, the conclusions, I think that tell me maybe I am wrong, but the theoretical matrix performs well because you have syntactic data, and you, in such a way, you perform a model that follows that theoretical model. So it is reasonable. It's expect that the theoretical matrix performs well, but it's also a good news that empirical metrics, metrics works well also. And the tree mix, I didn't really understand because the change of scale. I understand that the variance is different, but the center parameter, the mean of the matrix, I didn't really understand why is this shift, because it's a way to estimate the same covariant matrix. So yeah. Okay, different variance I accept, but different center parameter, I mean, is quite surprising.
Yeah, that is true. I'm not sure why it would be it would show a different mean.
Okay.
Because so in the trimix method, expanding a little bit on that, when you do the trimix method, when they estimate the W matrix, this parameter is also included. But in the case of the theoretical matrix, it was not included. In the case of the empirical matrix, it was not included. And in the case of the kinship matrix, it was not included. So we would expect a shift in variance. But now that you say it, I'm not sure why we should see a shift in the mean. I'm not sure about that.
Okay.
Yeah.
Maybe Fernando.
I have
has-
I mean, it's maybe like a suggestion. But like, I mean, it might require you to modify the tree mix code, but you know, there's this like arc sign transformation that you can make to the allele frequency so that they have sort of equal variance. You might be able to get rid of this nuisance that comes from the tree mix estimation by doing that. I mean, in the tree mix paper, they mention it, but they don't actually implement it. It might be a sort of way to better correct.
In the tree mix method, they say that they don't implement it because it actually increases the effect of low frequency alleles. So I guess it makes sense because.
Well, but I mean, is there a reason why, like, I mean, you're sort of weighting each snip the same, right? I don't know, that doesn't seem intuitively wrong or right. It's just a different way of.
Yeah, but in this case, you are also assuming that there is no mutation. So in principle, you should, yeah, I don't know. Yeah, maybe it could work, yeah.
Okay. I have
Regarding,
no more questions.
I think that you mentioned,
Mm.
you mentioned something before we spoke about this, but yeah, I don't remember.
I don't remember also. I don't know.
Oh, yeah, I remember. Yeah, the trimix, sorry, the theoretical matrix performs better.
Ah, okay.
Obviously, yeah, because I specify all of the parameters, but the results shows us two things that are related really. When we see the ROC curves. Here, sorry. So the theoretical matrix performs better, and then the other matrices do not perform very well. I mean, this is expected. The theoretical matrix is like the best that you can get. So how bad it works depends on how bad HFLK performs in this case with the theoretical matrix. But when we see the estimations of the covariance matrices, how bad they work is both for estimation of demographic history or covariance structure and because of HFLK's performance. So basically what we see is that we are actually evaluating the performance of the evaluation of the covariance matrices when using HFLK and not just the performance of HFLK under admixed populations.
It's okay. That's right. Thank you very much.
Thank you, Paula.
Okay, thank you. One question I have has to do with sort of the beginning, how you treat allele frequencies. Okay, and you know, there's known distributions of allele frequencies under the Wright-Fisher model, at least for single populations. And you sort of do a couple of things with that, in terms of your assumption of the distributions, how they compare with the Griffith, results for the allele frequency spectrum, and also sort of cutting off the low frequency alleles. So I want you to comment on that and how that may affect your departures from the baseline expectations, affect the outcomes.
So for the last question, the filtering of the low allele frequencies, that has something to do what we discussed. The model assumes that there is no mutation. So if there is mutation, you would expect to have very low allele frequencies at some loci. So with this filter, you take those mutations out of the question and only take into account old mutations that have already been present in the ancestral population. So that would fix that. And it is also the maybe inertia or custom of filtering out the low frequencies because of genotyping errors. And I don't know if I understood correctly your first question. Can you repeat it?
Your assumption distribution of the remaining alleles, okay, relative to the allele frequency spectrum. Okay. How do you treat then the remaining alleles, the one that you don't cut off because of low frequency error concerns? Okay. You assume a normal distribution of those, or that's
yeah, you assume that the allele frequency spectrum. I don't make any assumptions about the distribution of the allele frequency spectrum. The assumptions that I make is how they evolve how they change through time. So you have a frequency, the next generation, it will change with a normal distribution centered on the ancestral allele frequency. So the assumptions are not on the allele frequency spectrum, but on the evolution in time.
Yes. The other question is at sort of the other end. Okay, let's say you go ahead and apply your the test to real data, and as you well show in your thesis, there's conflicting or confusing situations because reality is a little more complex than any models we can think of. How do you consider additional types of data or information from, say, real data, sequence data, and all that, to sort of put your results into context? Okay, you come up with a significant result for a particular region or gene. Okay, what are the things you should do in terms of checks and balances to make sure you're on the right track and not just have a false positive? Okay.
Yeah. So first of all, I would what I would do is take famous data, for example, testing, I don't know, previous studies that have worked with hierarchically structured populations, such as sheep or some other population that evolves in a tree-like manner, and try to detect the same regions that have already been detected by other methods. For example, methods that are not based on population differentiation, but are based on a single population or whatever. See if I could basically reproduce the same results. And after that, what I would do is, if I get a new data set, for example, and if I get a positive, let's say, I would do like an ontogeny analysis. I mean, I would see what's the genomic region what's in the genomic region that's in the positive. How can I explain if there's a gene, if there's a regulatory region, if we can see what's the history of this particular genomic region, all of this information that can be accessed either through public data or through inference in the particular genomic region. That would be what I would do.
Okay, any other questions from the members of the jury? I take it as a no. Thank you. Now we're going to take questions or comments from the advisors. Okay, maybe Mindy, you want to start?
I wasn't prepared for.
You just pass the.
No, I can just pass through. There's Hugo and Bertrand also, but yeah, as Paola already said, the work we really enjoyed working with Gastón, because he really has a lot of skills in different branches of science. So the interdisciplinary with Gastón is really interdisciplinary, but all in one. So it's really easy to work with him. We talked a lot about statistics, but as well, he performed really good programming and the next flow. So it was like really clear, and he has his own ideas. So it's not always one directional, but it's really discussions with somebody that really understands what he's doing and thinks a lot about that. So I think that you already saw that in the presentation and in the answering of the questions. So yeah, that's the comment.
Thank you.
You're welcome. And I don't know if Hugo or Bertrand wants to say a few words.
Just a couple of words. Gaston is really nice guy, and the amazing thing is, in fact, he has done 2 theses. He changed of subject in the middle, but before changing, he finishes the other one. Yes, he's not present because he want to stay in population genetics, but the other is basically in bioinformatics. He finished the work and started a new one, all in the same period. So I think it's fantastic.
Thanks.
I can say a few words. So I followed Gaston's work from far, and far in space and like sporadically, I would say. But I found that he really did a great job, and I really developed this empirical approach to compute the covariance matrix, but I had no idea whether it worked or not. I just said we could do that and just implemented it, and then Gaston really took the thing, and now I'm really happy that he tested it and he showed that it works. So it's also very good for the software, and he did a very great job. And I think he also answered very well to the questions I found, and he understands the FLK model very well, and it's very nice also to see other people. When you develop a model and then you have somebody that comprehends it and uses it and improves it, it's also very nice to see. So thank you, Gaston, for that.
Thank you, Bertrand.
So
now I don't know if anyone has any questions, but I would like to thank you all for the few slides. So excuse me, but I will start to speak in Spanish, and when applicable, I will speak in English. So, primero que nada, muchísimas gracias trabajar en esto me encanta y no sería posible si no estuviera un montón de gente al lado mío. Primero que nada, mi familia, mis padres, mis hermanas, mis tíos, mis abuelos, mis mascotas, todas estas personas o seres vivos que la verdad que están ahí y no sería posible trabajar si no tuviera el apoyo de todos ellos y ellas. Después también están mis amigos, amigos de la vida, amigos más recientes, amigos que siempre están por la vuelta o no, a veces aparecen, a veces desaparecen, pero que me apoyan todo el tiempo. My friends, love you all. Y mis tutores académicos, mis referentes, my supervisors, my reference, my colleagues, all of you that también son mis amigos, muchos de ustedes, y la verdad que no sería posible tampoco sin su apoyo. Y nada, y los chistes, todo. Quiero agradecer particularmente, bueno, a mi madre, que está acá por suerte, a mi padre, a mis hermanas, y también en especial a Luisa, a Natalia, a Maine, que fueron de tremenda ayuda en esto que contaban de que cambié de tesis, básicamente. Ese fue un momento muy estresante para mí, y nada, y en todo momento ellas y todos los de la UBI en realidad me contuvieron pila, y la verdad que lo agradezco porque no hubiera sido posible seguir adelante con felicidad, digamos, sin su presencia. So I also want to thank Alexandra and because we're really good friends and we text each other a lot. Well, she texts me, she sends me like documents, but I don't really text her much. It's a one-way kind of relationship. So yeah, I really want to thank her. She's a real hero for science and for society in general. So yeah. And a riesgo de parecer un auto de Fórmula 1, voy a agradecer a todas las instituciones. I want to thank all the institutions that have allowed me to do this work. Quiero agradecer a la Universidad de la República, que me formó desde el bachillerato, desde la licenciatura, al Instituto Pasteur de Montevideo, que también me formó desde la licenciatura en etapas más tardías. Quiero agradecer a Cluster UI, que fue donde hice un montón de estos cálculos. Varios mails me mandaron pidiendo que lo use bien y lo ignoré bastante, pero está. Quiero agradecer al departamento de genética, que trabajé un montón de tiempo ahí y que, bueno, ahora lo tuve que dejar prematuramente por un tema que me tenía que recibir y que ahora me voy para estudiar al exterior, pero bueno, les quiero agradecer porque nada, fueron años formativos también como docente, como investigador y como persona también. Y bueno, quiero agradecer por los fondos, lo que me paga la comida y el apartamento, que son la CAP, PDCIVA, que me dio esta computadora, y también otros fondos para congresos, etc. ACICADA, que también me permitió trabajar como docente y trabajar como investigador también. A la ANI por darme una beca para trabajar en la Universidad de Florida durante 6 meses en el contexto de mi maestría anterior. Y quiero agradecer también a Facultad de Medicina, a Facultad de Ciencias y a Facultad de Ingeniería por todo lo que me dieron. Así que bueno, muchas gracias a todos y todas. Todos.
Bueno, ahora los tutores y el jurado nos vamos a otra sala, pero el resto se pueden quedar charlando en esta. Así que nada, disfruten el tiempo con Gastón. Vamos a pausa.
Maine, I think you're muted. Are you talking?
Yeah, you're muted.
Yeah, sorry.
So I'm going to do this in Spanish. Muchas gracias, Gastón. Queremos felicitarte por la versión escrita y la defensa oral de la tesis, la respuesta a las preguntas. Se refleja más un comando claro del tema, lo cual es sumamente importante, y un conocimiento de los fundamentos, que es siempre lo que te va a permitir proyectarte hacia adelante. Así que te felicitamos. Por supuesto, damos la tesis por aprobada. Y veremos cómo hacemos para la colección de las firmas del acta en forma remota, pero solo de forma remota. Así que muchas felicitaciones y creo que es unánime de parte del tribunal y de todos los tutores también. Y nos congratulamos con tener una tesis y un tesista de tal excelente nivel.
Muchas gracias.
Que te va a tener en los próximos años, tengo que extrañar acá.
Muchas gracias. Gracias al tribunal también.
Director.
Muy bien.